Thanks very much for that introduction. So you've summarized what we're all about. So programmable cell therapies. So wouldn't it be great if you could develop a cell therapy that wasn't just designed to perform one function and one function only, but was actually designed to respond to the body, to respond to the triggers, the disease state, to respond to specific conditions. Now, we've been trying to do this. If you think synthetic biology principles, you build parts. People have tried to build genetic circuits, build logic gates into these circuits, use lentiviruses, various delivery systems to drop these systems into the cell and hope that basically those circuits play out as designed. Now, 30 years in the field, we know so much of biology when you actually put it into a cell setting or an in vivo setting, it all conspires against you. Epigenetic silencing happens, instability happens, immunogenicity of your constructs happens, a whole series of lessons we've learned for why this doesn't actually work in reality. So the approach that we take is to think totally differently and say, well, actually, if we look at the cell orchestrates its own gene expression in highly exquisite ways. It's incredible the way that we have the same chromosomes in every cell of the body, but radically different function of each individual cell. And we now know through our analysis and understanding of the 98% of the genome that is not coding for structural proteins, but is regulatory elements. We've discovered a whole series of layers of gene regulation. MicroRNAs is one of those, and everyone who follows the Nobel Prize obviously knows that won the Nobel Prize in Medicine last year. So microRNAs are the vehicle that we take. So rather than trying to edit in a expression cassette or a construct into the cell and hope that it performs, we say, well, let's hijack the cell's own gene regulation system and subvert it to our own purpose. So we don't have to put an expression cassette in. We don't need to worry about promoters, transgenes, UTRs. We basically go in and we identify a microRNA that's produced by the cell under a specific set of conditions, and we recode that microRNA so it now no longer expresses its natural innate function, but it's been reprogrammed to silence a gene that we want to have silenced under those conditions. And hopefully you'll see the logic more as I go through it, and I'll give you some examples of where we're focused. So the company now, we're 3 years old. It's based on, we spun out the technology from actually a plant company called Tropic Bio, who very imminently are about to launch a non-browning banana, which is, so how does that relate to human therapeutics? Well, the basic system of gene regulation translates into human cells. We went out with this as a hypothesis, said it works in plants, therefore it should work in humans. And over the last 3 years, we've established the platform, we've humanized it, we developed the team, we've industrialized that platform, and we've demonstrated now with data. And I'm very short on data because this is a very quick presentation, but we have a whole body of data to show that that works. So we've built a strong team. This is my fourth CEO role, so I've done many things. I've exited 3 companies, 1 through IPO, 2 for sales, through sale, and the rest of the team is all veterans in this field as well. We developed a very clear strategy of where our therapeutic focus is going to be. So far, we've raised $13.5 million to support building the team and building the function. And we're now, as a seed round, we're now at the stage of doing a seed extension because we're at the cusp of generating pivotal data in an in vivo setting in 2 of our lead programs. On the basis of that data, that's going to take us into candidate selection by the end of this year, and we'll be doing then our Series A next year to support our non-clinical and then on into the clinic. So of the 6 to 8 million I say we're looking for here, so far we've got commitments of 5.8. So we've got a little bit more of a squeeze to do. So if anyone's got the odd million in their back pocket, then I'd be very happy to talk to you. But we're close, and it's looking, all of our investors are supporting us. You saw our investors on the previous slide. Lilly Ventures are on board, a series of EIS funds from the UK are on board. So programmable cell therapy. So out of everything that we could do, we've decided to focus on solid tumors because we all know from the CAR-T story, we've seen huge success in liquid tumors, but we've seen huge resistance in the translation of that to tackle solid tumors. And we know that the solid tumor environment has a very distinctive fingerprint of microRNA expression in the cells that migrate into that tumor microenvironment. So it's a really good target for us. And so we basically got a product pipeline that is 2 ex vivo cell therapy programs, one in CAR T, one in macrophages, which we love macrophages. We've also been working on hyperimmunogenicity platform that's been applied to type 1 diabetes. That's up for partnering. So in type 1 diabetes, our approach there is to link the silencing of the MHC and HLA genes to cell identity so that you can take an iPSC cell, differentiate it into a pseudo-islet cell, but it's only the terminally differentiated cell that's hyperimmunogenic, because it's only in those differentiated cells that the microRNA that now does the gene silencing of the MHC complex actually kicks in, and we can get tunable silencing to avoid the T cell adaptive and the innate T cell and NK cell response. And then we also have programs that we're conceptualizing as an in vivo editing approach. And here CMT1A is a classic example. So this is a disease where you have a gene duplication where too much expression causes the disease. You can't knock it out because you cause another disease by having too little expression. So tunable silencing is key. So we can, because our microRNAs can be programmed to have tunable silencing effect rather than just a knockout effect, we can tune to the right level through an in vivo approach and hit the sweet spot of gene expression. And that can't be achieved by siRNA approaches or classic RNAi approaches or by knockout through gene editing. So our platform provides these very unique set of capabilities that are distinct. It's sort of an amalgamation of the best of gene editing and the best of siRNA gene silencing into a unique platform where we can achieve with recoded microRNAs programmable, stable, specific, tunable, and multiplexed silencing. So by going into a single edit at a single allele, we can actually have identified a way to actually tunably silence multiple genes. And that's key for our T cell program. You can imagine there's a huge amount of tech stack building that's gone behind this. It covers from the bioinformatics, the design, taking massive libraries that we can screen now up to 6,000 unique microRNA combinations can be built into a library. We can test that in vitro models. We can deconvolute all of that to get to the ideal constructs to take forward into engineered cell lines. We've roboticized all of that as well. So very simply on the CAR T side, what we've shown that we can do is reprogram a microRNA to get checkpoint pathway inhibition in a way that is linked to T cell activation. So rather than doing a knockout of checkpoint pathways, what we can do is when a CAR T cell engages and is activated, microRNA gets switched on, which gives a tunable silencing of a checkpoint pathway or multiple checkpoint pathways. And so we can do this through a single edit. So you don't end up with an over-engineered, exhausted T cell by the time you get to a patient. You can develop a tunable T cell that is activated in an on-tumor manner. And already running late on data, so we do have data on time, sorry. We have data to support this, and we're taking this forward into in vivo studies over the next couple of months. On the macrophage side, then, what we want to do is two things here. One is to be able to silence certain pathways or within the macrophage selectively in the tumor microenvironment to silence pathways that influence the polarization of those macrophages from a TAM state to a pro-inflammatory state. And so we validated a number of targets within the macrophages through knockouts that we know that if we knock out those targets and expose those macrophages to the tumor microenvironment, the context of the tumor microenvironment, we can still maintain the pro-inflammatory phenotype of those macrophages. And so we've now can multiplex silencing of a number of those pathways by editing microRNAs to achieve that. and then put those into in vivo models. We've now demonstrated it in a mouse in vivo model where the polarization takes place towards a pro-inflammatory state, and we're following that up with more in vivo data over the coming 3 months. So again, if the vision is to create a therapeutic, a cell therapy that rather than being a once and done and hope for the best, is an adaptive cell therapy that responds to the disease state of the cell, that's what our platform is capable of delivering, and we're exemplifying it in these 2 oncology programs.